Video number 13, we continue talking about lipids and more precisely their classification. Lipids may be classified into two broad types, the saponifiable lipids and the non-saponifiable lipids. So the saponifiable lipids are the lipids that can be used to make soap reaction or saponification reaction. What is saponification reaction? It is the addition of a base, sodium hydroxide for instance, or potassium hydroxide, with the lipid that contains fatty acid or fatty acids. The result is soap. The soap is the salt of the fatty acid plus water. This chemical reaction is well known. It is the reaction of saponification, base plus fatty acid. It gives a soap. The soap is the salt of the fatty acid plus water. The lipids that are saponifiable are triglycerides, phospholipids, glycolipids, and ceraids. These are the different lipid types that may be saponifiable because they contain fatty acids. Without fatty acids, saponification cannot take place. Since a fatty acid is important, let's first start by defining a fatty acid. A fatty acid is a molecule that is amphipathic, sparingly free, rarely free. It is rather esterified by glycerol or linked to sphingosine. We rarely have fatty acids that are free inside the cell. They are linked to glycerol by an ester bond or linked to sphingosine by an amide bond. This is a detail. This is the general formula of a fatty acid, CH3CH2 N times COOH. N, the number of CH2 here, N ranges between 2 and 20. Therefore, the total number of carbon atoms per fatty acid molecule is 4 to 22. They are amphipathic because they have a hydrophilic head, which is the carboxyl group, and a hydrocarbon tail, which is nonpolar, which is hydrophobic. An amphipathic molecule is a molecule that has a hydrophilic head plus a tail that is nonpolar, that is hydrophobic. The hydrophobic tail may be saturated or not. It may contain 1 to 6 double bonds. If the fat substance is rich in saturated fatty acid, it is rather solid at room temperature. Example, animal fats, most animal fats, butter. These are rich in saturated fatty acids and they are solid at room temperature, while the plant-derived fats, they are liquid at room temperature because they are rich in unsaturated fatty acids. The unsaturated fatty acids are those that contain at least one double bond. Of course, unsaturation of fatty acids will have a direct effect on lipid fluidity in the membrane. Fatty acids are highly energetic. They are oxidized in mitochondria and in peroxisomes. This topic will be described in chapters 9 and 10. So we postpone this idea to chapters 9 and 10, where we consider oxidation of fatty acids. Now just remember that fatty acids are energetic molecules. They are used by the cell to produce ATP, to produce energy. So this is the description of fatty acids. Let's go back to the saponifiable lipids and the different types. The saponifiable lipids are they contain fatty acids, of course. Fatty acid, if the fatty acid is linked with the glycerol, it is an esterification reaction. One glycerol molecule having 3 hydroxyl group can make ester bonds with 3 fatty acids. So this is a glycerol. This is an alcohol group, a hydroxyl group, reacting with the fatty acid. It is a reaction of esterification. Refer to the chemistry course to define esterification reaction. The ester bond is COOC. One ester bond with one fatty acid, it is a monoglyceride. Glycerol plus one fatty acid, monoglyceride. Glycerol plus 2 fatty acids, diglyceride. Glycerol plus 3 fatty acid, it is a triglyceride. So this is These are examples of saponifiable lipids. Of course, the most common one is the triglycerides. Sorry, because triglycerides are family, R1, R2, R3 are different types of fatty acids. Let's consider some examples of fatty acids that can be used. We have butyric acid, myristic acid, palmitic acid, stearic acid. You are not supposed to memorize the numbers. Just remember that these are examples of saturated fatty acids. They may occur in triglycerides, especially triglycerides of animal cells. Regarding the fatty acids that are unsaturated, we have three main examples: oleic acid and linoleic acid. These are abundant in olive oil. And we have arachidonic acid, which is also unsaturated. It is present in animal cells also. So here are examples of fatty acids that may form triglycerides. Of course, triglycerides are diverse. I repeat, don't memorize neither the number of carbon atom per fatty acid nor the number of double bonds. Just remember the examples of saturated, 4 examples of saturated fatty acids, and the names of 3 examples of unsaturated fatty acids. The triglycerides is the first family of saponifiable lipids. The second family of saponifiable lipids is the family of phospholipids. And this family of phospholipids is subdivided into glycerol-derived phospholipids and sphingosine-derived phospholipids. The glycerol-derived phospholipids is made up of diglyceride plus phosphate. What is a diglyceride? I remind you that a diglyceride is a glycerol making 2 ester bonds with 2 fatty acids. This is the first ester bond, COOC, second ester bond, COOC. So a phospholipid that is derived of glycerol, it contains a diglyceride plus 1 phosphate. Okay, the figure is represented here. We have glycerol, the purple box, 2 fatty acids, R1, R2, okay, 2 different types, and the third hydroxyl group. Which third hydroxyl group? This one. The third hydroxyl group, instead of linking a fatty acid, in case of triglyceride, there are 3 fatty acids. Triglycerides are neutral fats. However, here for the glycerol-derived fatty acids, instead of the third fatty acid, here we have a phosphate. So these are phospholipids that are derived of a glycerol. We name them phosphatidic acid. Are they neutral fats as triglycerides? No. Triglycerides, here also another schematization of triglycerides, glycerol plus 3 fatty acids, 3 ester bonds, COOC, just a different representation of triglycerides. Another representation of triglycerides, this is a glycerol in the box, and these are the 3 fatty acids that make 3 ester bonds. It is exactly the same concept as here, just the shape is slightly different. So here the molecules are not neutral. Here the triglycerides are neutral. The phospholipids are not neutral because this phosphate group is an acidic group which is negatively charged. Therefore, here we have a hydrophilic head and we have a hydrophobic tail region here. This is the region where we have 2 tails that are hydrophobic. Clearly, the hydrophilic head is polar and the hydrophobic tails are non-polar. Do we have a single type of phospholipids? Of course no, because already we have different phospholipid types depending on the fatty acids that are linked here. Depending on the fatty acids, we have different phospholipid types. But also we have different phospholipid types because other molecules are added to phosphate, such as ethanolamine. The blue box is ethanolamine. You are not supposed to memorize the chemical structure, don't worry. When we have ethanolamine that is added, now the phospholipid is different from the first one. It differs by the hydrophilic head here. It is named phosphatidyl ethanolamine. Phosphatidyl, this part, the part here is phosphatidyl added by ethanolamine. We might have choline, the green box. It is phosphatidyl choline. We might have inositol. It is phosphatidyl inositol. We might have serine. It is phosphatidyl serine. So these are different types of phospholipids, all of them derived of glycerol. They differ by the fatty acids, but also they differ by the hydrophilic head part. All these phospholipids are amphipathic, having a hydrophilic head that is schematized as a circle, and hydrophobic tails that are schematized as a zigzag vertical line. These phospholipids are abundant in the membranes. Remember in this figure, in this table, I showed you different phospholipid types: phosphatidyl ethanolamine, phosphatidyl serine, phosphatidyl choline, etc. I repeat, you are not supposed to memorize the chemical structures. However, you must be able to say that phosphatidyl inositol is equal to 2 fatty acids plus 1 glycerol plus 1 phosphate plus inositol. These data are included in the name already. Phosphatidyl choline includes 2 fatty acids, 1 glycerol, 1 phosphate, 1 choline. Of course, I mean phosphatidyl choline that is derived of a glycerol. Okay, fine. So these different phospholipid types occur in different membrane types in different proportions. These differences in terms of composition provide its membrane, its unique characteristics, and its unique functions. Let's move now to the other type, the sphingosine-derived phospholipids. The sphingosine-derived phospholipids are made up of ceramides plus phosphate. Let's define a ceramide first. A ceramide is a sphingosine, this part which is included in the box, the purple part plus this part. This is the molecule of sphingosine. Don't worry, you are not supposed to know its chemical structure. This is the chemical structure of sphingosine. This hydroxyl group is this one. This amino group is this one. This OH is this one. Okay. And this CH2 is this one. Sorry. Okay. So a ceramide is the sphingosine plus a fatty acid. So a phospholipid that is derived of sphingosine, it is equal to sphingosine plus fatty acid, making a ceramide. The ceramide plus phosphate, it makes a phospholipid that is sphingosine derived. And again, we have exactly the same thing that happens. We have addition, for instance, of choline. This will be phosphatidyl choline, phosphatidyl serine, phosphatidyl ethanolamine, etc. So these different phospholipid types, they may be derived of glycerol, they may be derived of sphingosine. Globally, it is the same amphipathic structure with a hydrophilic head corresponding to this part and hydrocarbon tails that correspond to the hydrocarbon tail here and the hydrocarbon tail that belongs to sphingosine. The sphingosine-derived phospholipid are abundant in the brain, for instance in the myelin sheath, sphingomyelin. Sphingomyelin is rich in sphingosine-derived phospholipid. What is the main advantage between, I don't know, it is only an advantage. The main difference between sphingosine-derived phospholipids and glycerol-derived phospholipid is a difference in terms of stability. Here we have amide bonds that are much more stable than the ester bond that occur in glycerol-derived phospholipid. This is a detail. You will not be asked any question about this point. I mean about stability of the bond, about the difference in terms of stability. Moving to the third type of saponifiable lipids, glycolipids. We will talk about glycolipids, but now we have to pay attention to an important point. In glycolipids, we have also the possibility to have sphingosine derived as well as glycerol derived. These glycolipids, as the name clearly indicates, they don't contain phosphate. In the previous name we had phospholipids, here we have glycolipids. So glycolipids contain, instead of phosphate, which phosphate? This one. Instead of the phosphate in the orange box, we will have a sugar. Instead of phosphate, we will have a sugar. A sugar may be monosaccharide or disaccharide or an oligosaccharide. These glycolipids, again, they may be derived of sphingosine, but also they may be derived of glycerol. This is glycerol. This is glycerol. This is glycerol. The two fatty acids, they are unsaturated here. A disaccharide, a monosaccharide. All glycolipids, whether derived of sphingosine or glycerol, they have amphipathic property with a hydrophilic head schematized by a circle corresponding to the sugar part, and the hydrocarbon tails corresponding to the fatty acid tails in case of glycerol derived phospholipid, corresponding to the tail of a fatty acid, and sphingosine in case of sphingosine derived glycolipids. These glycolipids, unlike phospholipids, they are present only at the outer surface of the plasma membrane. We don't encounter glycolipids at the inner side. And the most poignant example is the example of glycolipids that form blood groups. You remember that the blood groups are made up of, I'm sorry, I have to move to one of the first slides, this one. The oligosaccharide, I explained in a previous video, this oligosaccharide makes the antigen OA, antigen O, and antigen B. This belongs actually to a glycolipid and sometimes to a glycoprotein. Let's now be interested in the oligosaccharide that belongs to a glycolipid. Clearly, the antigens must be exposed outwardly, outside the cell. This is why I'm saying that the glycolipids, in case of blood group antigens, but in all other cases, glycolipids are found only exclusively at the outer membrane. They may form up to 5% of the outer lipid layer. They are not as abundant as phospholipids. Many other functions depend on the glycolipids that are present at the cell surface. For instance, recognition between two cells sometimes involve glycolipids. Glycolipids can be subdivided in a different manner other than sphingosine derived, glycerol derived. We can divide the glycolipids into cerebrosides and gangliosides. Cerebrosides contain here a simple sugar, a simple monosaccharide such as glucose, such as galactose. However, the gangliosides contain more branched oligosaccharide with the N-acetylnuraminic acid. I explained this name earlier in this set of videos. The galactocerebrosides, why the name cerebro relative to the cerebellum or to the brain? It is because the galactocerebrosides are extremely abundant in the nervous system, in the outer lipid layer of the nervous system cells. Here is an example of ganglioside. All this part is an oligosaccharide that is quite complex, and these are the two fatty acids. This is the fatty acid, this is the sphingosine tail, and we have a sphingosine derived ganglioside. These are examples of glycolipids that are glycerol derived. I repeat, you don't have to memorize the chemical structure. you must be able to conclude from the name the composition. If I tell you, for instance, a glycerol derived glycolipid contains, you must be able to say it contains 2 fatty acids plus 1 glycerol plus a sugar molecule or 2 sugar molecules or 3 sugar molecules, etc. A sphingosine derived glycolipid contains 1 sphingosine, 1 fatty acid, and 1, 2, or 3, etc. Monosaccharides. Sometimes we have heteropolysaccharides, of course, it is possible also. We might have modified sugars, I mean. The last family of saponifiable lipids is the family of cerides. Cerides are actually special lipids containing a fatty acid plus an alcohol that has a long hydrocarbon tail, named fatty alcohol. When an alcohol has a long hydrocarbon tail, it is a fatty alcohol, such as this one. This one, we have CH3, CH2, 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 etc. CH2OH. This is the cetylic alcohol. And this is the palmitic acid, a fatty acid. When they form an esterification reaction, they result in a ceride. Cerides are lipids that are present in beeswax. They are present in the cuticle of plant leaves. You know that the plant leaves are covered by the cuticle, which is glossy and shiny, sometimes relatively thick. This glossy and shiny cover of the leaf is made up of cerides, but also we find ceride in cork, natural cork that is produced by plants, of course. All these are impermeable, actually. They provide impermeability to the leaf, etc. In the next video, we consider the non-saponifiable lipids, inshallah.